Hello, welcome to the Excited Education channel, the YouTube channel which brings you about mathematics and biology. I'm teacher Amy Chris. As usual, now we are going to discuss about biology on the chapter called viral replication. Hello, welcome to the module. What is viral replication? Let us see different chapters that we likely to look about on the viral replication. What I have to tell you, if we say viral, think virus. Replication, think about its development and its division. Here we will see the general aspect of viral replication, where we will find that there are different stages that are involved in viral replication. Another point we likely to see is what we call replication of bacteriophage or phages, where in this replication of bacteriophages we are likely to find lytic phage replication comprises with T-even phage replication of RNA bacteriophage, and we will see what we call methodology assaying for lytic phage, and the bacteriophage growth curve temperature phase, what we call lysogeny, and we will see as subtopic number 3, which is called replication of plant virus, where we will see the historical perspective. Plant virus show RNA can serve as the hereditary micromolecule. Let us see. Go ahead. And the last chapter, which is called the replication of animal virus. If we finish to see the replication of the plant virus, we go to the replication of animal virus, where we see the methodology they likely to use in laboratory culture of animal virus, and we will see the replication of DNA animal virus, replication of RNA animal virus, and we will see also highlights how new viral disease originate, transformation of animal cells, historical perspective, and discovery of oncogenes. So here, allow me to start and see the general aspect of viral replication. What are the general aspects of viral replication? So here, as you see, we have seen as a first point on the chapter outline. Then here, general aspect of viral replication, there is what we call virology. Virology is general a term that are likely to be used in the study of virus. So viruses are cellular, non-living microorganisms that are totally dependent on host cells for replication. You know that viruses are the microorganisms. They cannot be seen with our naked eyes, just we use microscope to view their parts and their shapes. Totally dependent on host cell for replication. They depend on another host or another living organism in order to do their replication. These are general aspects or general characteristics. Then premises for a virus to complete replication in host cell. Here we will see the specificity and the permissibility in the compatibility. We are going to differentiate these three words. What is specificity, what is permissibility, and compatibility. Specificity is based on the ability of the virus to physically attach to the host, the ability to attach to the host, and the ability of the viral nucleic acid to direct viral replication within that host. For instance, we have the virus in its host. That ability, the function or the system that is likely to use to be attached on that host is what we call specificity of the virus. And permissibility means the host cell must allow entry and must not degrade the viral nucleic acid genome. This is what we call permissibility. Permissibility is permeable. Permeability is allowing the passage of substance. Certain substance means that in the host cell they are likely to open their cell membrane and allow the viral nucleic acid to enter, and it's not likely to degrade those host cells. So this is what we call permissibility, meaning the host cell must allow into and must not degrade the viral nucleic acid of genome. Yes. Then compatibility simply means that the virus must be capable of using and directing the metabolism of the host cell of the viral replication. Here, in compatibility, we find that the virus it have a capacity of using and directing the metabolism of host cells. Simply means those reactions that are likely to take biological reactions that are likely to take place in the body of the host. The virus is in charge of controlling those reactions that are likely to take place in order that it help their viral replication. You know that if they are likely to replicate while they are inside of the host, simply means their activities or their cellular activities as a virus, they are likely to be used as that of host. The more the cellular activities of the host are acting properly, the more the viral activities of the virus working properly. Means its replication of the virus, it depends on how the activity is being done on the host cells. So here, these are three words that are likely to understand in the general aspect of virus. We find that it is permissible for virus to complete replication in host cell, while we find the specificity, permissibility, and the compatibility. So let us see the stages of viral replication. What are the stages of viral replication? So viral replication is the duplicating of the virus strands into new ones. So here, there are at least 6 stages of viral replication. So here, adaptation, there is what we call adsorption, penetration, uncoating, synthesis, assembly, and release. We are going to explain one by one according to how it is steps by step. So let us start on the adsorption. Adsorption, virus attaches to the outer surface of suitable host cell at specific binding site. Here, first, this virus have to attach on the outer surface of the host cell at the specific binding site. It's likely to bind on the specific site, but not anywhere. Where it's likely to be bind is where the cell is ready to be attached of virus. So, if virus attached to the specific cell site, host of the cell, yes, host cell, so it is what you call adsorption. So, for penetration occur when the virus or its nucleic acid genome crossing the plasma membrane and enters the host. Then first, we know that adsorption occurred when the virus is coming and attached on the surface of the host cell in the presence of binding site. So after it engage or it penetrates through the membrane of the cell of the host and then move into inside all the cytoplasm of the cell. So here it occurs when the virus or its nucleic acid, those components of the nucleus where I find the nucleic acid and the DNA and RNA. So they are likely to be occurred or they are likely to be crossed up through plasma membrane of that host cell and enters into the host. So this is what we call penetration. So what we call uncoating. Uncoating is when the viral nucleic acid is released from the capsid in virus that replicates within the bacterial cell. Penetration and uncoating usually occur simultaneously. Viral nucleic acid genome enters the cell. So here, if the cell reached in the cytoplasm of the virus reached in the cytoplasm of that host cell, it's likely to divide and release what we call capsid, and in virus that capsid likely to be replicated. Those replication occurred in the cytoplasm of the host cell, and this take place in the process what we call uncoating. So we likely to find that penetration and uncoating likely to be occur in simultaneous. So here viral nucleic acid genome enters the bacterial host cell. So as you see here, the uncoated, in uncoating, those nucleic acid genome enters the bacterial host cell. As you see here, these are the nucleic acid from the bacteria or from the virus. So let us see the next step, what is called synthesis. Viral nucleic acid takes control of cells metabolism to direct the synthesis of the protein. Here it is, if we say synthesis, you likely think about protein synthesis. So here, if the virus intervene in the cytoplasm of the host cell, it's likely to release those nucleic acid genome, and after it take part of the metabolic activities of the cell. It controls all metabolic activities of the host cell in order to allow its synthesis of the protein, and even it gets or it gains from it as the parasite. That's what we call synthesis. In synthesis stage on viral replication, it is the process where the virus likely to take a part in the metabolic activities and control the protein synthesis of the host cell. So let us see the next stage is what we call assembly. For assembly, we will find that other protein synthesis make a copy of viral nucleic acid. If you find that they have in process of other protein synthesis, it makes copy of viral nucleic acid. After the nucleic acid synthesis, let protein synthesis for a capsid production. For instance, here, if it reaches in the stage of assembly, there is what we call process which is called RNA protein synthesis, where it makes copies of viral nucleic acid. Those nucleic acid are likely to be copied in this process of assembly. Then after the nucleic acid synthesis occurred, where the red protein synthesis that is made for capsid reproduction. Let us see the next stage, which is called release. So after this, there is what we call in assembly, when the nucleic genome is packed into the capsid. So here in assembly, it is where the nucleic acid genomes is likely to be packed into the capsid. These are capsids of the viral cell. So in release, the assembly virus leave to the host. So after formation of its packages into the capsid, they are likely to be released out of the cell. So the viral replication over. So this is what we call stages of viral replication. We find that there is what we call adsorption, penetration, uncoating, synthesis, assembly, and release. I think we have heard more about this. So let us see what next. Let us see what we call T-even phage replication. What is T-even phage replication? This is the figure of T-even phage replication. The reason why it's likely to be called T-even phage, it's likely to be like having a T shape, a T letter shape. So structure T-even phage, the T-even phage have a capsid that has a distinct head and tail structure. This is this distinct head, and this is what you call tail fibers or tail structure. This is a part of tail, and this is a part of head. This is what we call T-even phage replication. So T-even phage, this is its magnification under the microscope. So this is what we call T-even phage. This is likely to be the head, and this is likely to be the head of the phage, and this is likely to be the tail. This is how it looks like in the cell. So and the entire even phage does not penetrate the bacterial cell, but rather the phage injects into it injects only its DNA into the bacteria. For T phage, it's not allowed to penetrate into the bacteria cell. It's likely to inject their DNA through those tails. They touch it on the bacterial cell and then inject those DNA into the bacteria. Then penetration and uncoating occur simultaneously for the T even phage. As we see in the stages of the viral replication, the penetration and uncoating is occurred also simultaneously on the T phage. Then colonized macrophage showing the attached of T even phage to the cell of E. coli or Escherichia coli and the extension of the tail tube as an injection needle. This is the structure of the T even phage as under microscopic magnification. So the T even phage, let us try to explain more about this T even phage replication, how it takes place and the process that is likely to be passing through. So T even phage has a capsid that has a distinct head and tail structure. These phages are designed by even numbers. For the example, T2, T4, and T6. That's why you call it T even phage. These are even numbers from 2, 4, and 6. These are what we call even number. Then T is their terminal. So this means that these T even phages, they likely to have designed by their even number. So and they have capsid which comprises tail and what you call head. So the DNA genomes is contained within the head, and the tail is involved in adsorption to the host cell. We know that in the stages of the viral replication, we include also the stage of what we call adsorption. So for the T even phage, they have DNA genomes and is contained within the head and the tail. Those genomes are likely to be involved in the process of adsorption as the stage of the viral replication. So the bacteriophage's tail release lysosomes. These are enzymes. So the tails of the bacteriophage, they likely to release the enzyme lysosomes, which allows breakage down a portion of the bacterial cell wall. You know that the cell wall is likely to be hard, and those hardness, it's likely to require the enzyme to be broken down. So this bacteriophage's tail is contained, or it releases the lysosomes, which allows the bacterial cell wall to be broken down. So the viral DNA is transcripted. So if the viral DNA is transcribed within the host cell, to make viral messenger RNA, the messenger RNA is subsequently translated at the ribosome of the cell to make a phage protein, RNA protein synthesis. This process occurs in RNA protein synthesis, as we see here. RNA protein synthesis, for instance, this is in this stage. We find that in RNA protein synthesis, make copies of the viral nucleic acid. Then this viral DNA, what you call comprises messenger RNA, they likely to translate at the ribosome of the cell to make phages protein. So here the T phage also have to know that under the control of viral nucleic acid genome, the bacterial cell synthesizes phage nucleic acid and various proteins that make up the capsid. This is what we call stage of later protein synthesis in the phage of the replication T even phage. So here under the control of viral nucleic acid genome, the bacteria cell synthesizes phage nucleic acid and viral protein that make up the capsid. Those viral proteins are likely to be used in the formation of the capsid in the process of the DNA replication or viral replication. Yeah, the head and tail are made up of protein coded by at least 5 and 3 phages genes respectively. For head and tail for the T even phage, they are made up of proteins coded. These are codings. Codings are those subunits that are likely to be used in the coding nucleotide to one another. So here, protein coded for they likely to use 55 and 33 phages genes respectively. So The head and tail units of the T even phage capsid are assembled, separated, and later combined. So they like to have divided or to be separated, head and tail, and after it combined, the release of T phage results in the rhizosis bacteria. If the phage if the T even phage is released out, they like to break the bacteria. Simply means if the process or the replication of the T even phage is taking place in the inside of the host cell, if the stage is complete, that bacteria is likely to be burst. So this is the process. As you see, this is the bacterial DNA inside of this one. After this transformed, this step what we call penetration. This is first stage, which is called the step 1, which is called attachment. Those T phages are likely to be attached on the bacterial DNA. After they release their genomes inside, then they take place into this. Take the second place, which is called the penetration. Those phage injected the bacterial cell, leaving the phage coated outside. Then the transcription. This phage DNA is transcripted producing phage messenger RNA, which is translated to phage protein. Then this allows DNA and the RNA to make their strands. Then the phage induced protein. These are the proteins that are likely to be made, which is called the phage induced proteins. In step number 4, the phage coated protein, other protein component, and DNA are produced separately. Host DNA degrade. You see, this is the stage number 4, and in this stage number 4, it is where it goes in stage number 5, what you call assembly. And in assembly, these phage components are assembled into mature virion. This is mature virion that is completely made. So this mature virion, they like to undergoes empty head. They have empty head. After the head, like to make the DNA inside the head. Those DNA inside of the head which are empty, they like to make a maturity stage. And if they reach in the maturity stage, they like to have strands, and the other nucleotides are likely to attach on their strand. Then after they undergoes complete maturity, and they form what you call head and the tail. This is head and this is head and this is tail. Then this is what we call those fibers likely to be like legs. So this is likely to attach on the base of the tail. Then the complete phage, even phage, is likely to be formed. So this is what we call the process of bacterial viral T even phage. So it's replication. Let us see the next. So also there's life cycle of phage T4. This is what we call life cycle of phage T4, and this is the process. As we see in the first stage, you can use that first diagram, or you can use this diagram. Also can explain more about the life cycle of phage T4. This you see here, if it attach the viral cell attached on the host, it likely to release the chromosomes. If there is their chromosome, the RNA messenger RNA made. Then this messenger RNA, if made in 2 minutes, the host DNA degrade, and then in 3 minutes the phage DNA made. After in 5 minutes the later RNA made. Then in 9 minutes they have tried to be matured, and in 12 minutes head and tail is made for this phage, and after 13 minutes the head filled. If this head is filled, the what we call virion formed. This is like to be the younger one of the viral formed. That's what we call virion formed. So in 15 minutes the what we call host cell rises. If cell rises, it releases those virulence. Then the complete stage occurred. So this is a figure. This figure shows the T phage adsorption and DNA injection. This is landing, attaching, tail contraction, penetrating and plumbing, and DNA injection. This is what we call T4. 1, 2, 3, 4. So what we call the T4 phage adsorption and the DNA injection. So here, this is a figure permission required reproduction or display. This is likely to be a cell, how it takes place. These are the T phages attaching on the host cells. This process occurred in this diagram. If those cells likely to occur, they use this, or they are attached in this way. These are the T phages. So there's a different parts of cell surface can be potential adsorption site. So these are how it is attached on the host cell. Let's check this. Let me zoom. So this is what we call bacteriophage, and also this is what we call bacteriophage, and we also can see this is what is a figure which shows how those process or those replication likely to be occurred. So T phage replication, this is also can use this figure to explain more about T phage replication. The assembly of T4 bacteriophage, not the sub-assembly line for the base plate. So tail tube and sheath, tail fibers and head. This is what parts that are likely to be formed in the process of the complete development or complete replication of the T4 bacteriophage. So you see this base plate protein. Yes, this is what we call base plate E, in the form of E. Then tube, likely to develop tube. Then here, in this stage, tube is developed. Tube and sheath, they develop what sheath. After they move in what we call whisker and the neck. This whisker neck is ready to come and attached on this tube and sheath, then to develop into forming this complete T phage. So this is the point between the tube and the whisker's neck is what we call cola, and those are tail fibers that are likely to be attached on this base. So this basal plate is where those tail fibers are likely to be attached on the T phage bacteria. Then tail fibers protein. These are tail fibers. Those fiber tails are likely to have tail fiber protein. Release of bird flu virus. These are what we call bird flu virus. How are released outside of the cell, and we find that if the T4 phage is complete, its replication is complete, the bacteria is likely to be burst in order to release those new cells that are already made. Replication of DNA. No, replication of RNA bacteriophage. The RNA phage has hereditary information contained in a single strand of RNA. This RNA designated as a positive RNA strand can also serve as a messenger molecule. So if we take the positive RNA can be translated at the ribosome of the bacteria cell to produce the protein for making the viral capsid. To make RNA viral protein, the positive RNA is used as the template for making the double strand of RNA that has both plus and complete negative strands. So this RNA bacteriophage replication, they like to form first positive RNA strand. After, it can be also served as messenger RNA. Those molecules are likely to be attached in presence of protein and making the viral capsid. After making those viral capsid, the positive RNA is likely to be used as template forming a double strand, where we find the negative strand and the positive strand. So they to make this replication form RNA nucleotide in association with RNA polymerase, they pair with the complementary nucleotides in the strand, and the RNA polymerase links them together. This is what we call RNA polymerase. They like to link the strand with their nucleotide. This is the enzyme that are required to take place in the process of RNA replication. Then the RNA strand is used as template for the transcription to produce new RNA copies. So a copy of plus RNA is then packed into a capsid and a new phage is assembled. After assembly, the host cell is raised by an enzyme called by the phage. You know that if the complete stage of RNA replication is already done, there is what you call rises of the host cell in order to release those phages. So this is a process of the reproduction of single-stranded RNA bacteriophage. We have starting with the RNA plus act as messenger RNA. They take an action of messenger RNA, then they what you call RNA replicase. They are likely to replicate in the presence of RNA replicase and form plus and negative RNA. These are replicative form. Then these replicate also to form the main RNA plus in process. This process what you call replication, and then after this they inject in the host cell. Then there is a process what we call viral protein and RNA genomes. Then these combined together to form what we call new viron. Then in this process what we call translation. And methodology assigning for lytic phages. Methodology for assigning for lytic phages. This is what you call the replication of lytic phages. Lytic bacteriophage cause the formation of branchi in RNA of bacterial cell growing on the agar surface. You think agar is the what you call alga. This alga is what we call assigning for lytic phages. This process is likely to take place in agar alga. They are likely to form what you call branchi in row of bacterial cells. So, dear students, allow me to stop from this point, and then we'll continue on the point what we call bacteriophage growth curve, and try to discuss about the curve or the stages that bacteriophage likely to occur in order to be complete in their replication through a curve. I'm teacher Amy Chris as usual. So I would like to make subscribe. Don't forget to subscribe on my channel to watch my videos that are likely to be followed. Thank you.